Many of you saw me yesterday, so I'm Les Bennett at UCSF. So we have 4 speakers today, and I will introduce them. The first is Eric Novik, who's Hurel's Chief Scientific Officer, and Eric is the lead developer of Hurel's primary hepatocyte co-cultures and Hurel Flow, organs-on-a-chip microfluidic platform, which is currently in development. Named on 3 of Hurel patent applications, in addition to leading Hurel's R&D initiatives, Dr. Novik also serves as scientific liaison to the company's customers. He received his Ph.D. in biomedical engineering from Rutgers University, writing his dissertation on the generation of adult liver-like cells from stem cells. So Eric, please join us.
Thank you, Les. It's an honor to have you on our team. A little bit of an overview of what Hurel does. We do co-cultured hepatic models. We use primary cryopreserved hepatocytes cultured together with cells of a non-parenchymal stromal cell type. These cells are put together and allowed to self-assemble, so it's really a self-assembled model. We have 5 species and 7 models that we currently offer. We have the Hurel human, the Hurel human pool, which comes in donors of 5 or 10, 15. Hurel rat, and then we have the Han-Wister rat. We do the Hurel dog, mini pig, primate, and mouse is under development. What we focus on is practicality and convenience as well as function. So we want it to be easy for our customers to use the plates or to receive samples. So plates are delivered by air overnight. They're ready for use after a brief acclimation period. You get the cells, change the media, put them in the incubator for about 4 hours, and they're good to go. Contract research is also available, and I think as I go through my slides, you'll kind of see that, and I'll talk about the different ways we've worked with our customers and how they receive data or do the analysis. So what we do is we deliver stable, enduring hepatic co-cultures. We have them in the human, just some nice pictures up there, primate, dog, rat, and we do them in any well or plate size that fits your needs. We can go from 384 to 6 well Petri dishes if it's necessary. So what we do is we identify a window of stable enzymatic function. So when we plate our cells, we keep them in-house for about 7 days, allow them to acclimate, kind of come back up to their steady state, and then we send them to our customers or start experiments. By that time, it's day 7, which we call customer day 1. So if you're looking at the bottom kind of graph over here, this is day 1, but in reality, it's 7 days since we plated the cells, and you just see this very stable kind of 3A4 function out to 18 days from when the customer gets it, which would be about, 25 days from when we plated the cells. And it could probably go out further. We just haven't had a need yet. If you have one, I'm sure it'll work. And we've done this for many, many of the enzymes out there. I'm just showing one here, 3A4. These cells form beautiful canaliculi, which goes to what Mark will be talking about from Optivia. We have functional canaliculi. This is 31 days out of culture from day 1, and this starts on day 6, and it's basically the same throughout. This is an example of a study that we did with UCB Pharma in Europe, and again, this is one of the working models that we have. So we incubated these compounds in our labs, sent them the supernatants, and they analyzed it with their bioanalyzers and sent us back this data. And it's really a good comparison of species-specific differences in metabolism or clearance. And what you see on the right is that for lorazepam, the dog in vivo clearance is very high, so it's 18.6 ml per minute per kilogram. And if you're looking at the data that we generated, it's about 85% metabolized in the first 4 hours, so very high metabolism. In the human, the in vivo clearance is very low, so 1.21 ml per minute per kilogram, and you could see that it takes 7 days just to get 66% clearance. So that was a good correlation, and the other part of that was taking a drug that was similar in both species, where you have the dog, which is 1.1, and the human, which is 1.6. This is ketoprofen. And again, you can see that it takes 7 days to get to 70% or 83%. And this is 7 days without changing media. So you add your compound to the cells. They sit there for 4 hours, 2 days, or 7 days. You stop the experiment. You take it out, and you do the data analysis. And Matt Huxter's going to talk more about this type of data. And another interesting thing that we saw was the differences in metabolite generation between the human and the dog. So you see all these nice peaks here that really correlate well to what goes on in vivo. This is a study that we did with another large pharma. This we did all totally in-house. We did all the work, analyzed all the data, and sent them back a report, which is another way that we can work. In particular, we did inhibition and induction, and here we're looking at a competitive inhibitor, classic competitor inhibitor, ketoconazole, and you see that we dosed every day for 3 days. And you see at time 0, so this is what, maybe 10 minutes after we dose, we call it time 0, and it's inhibited, and you see very little 3A4 function. Take it out for 72 days, and you still see the inhibited function. Now you remove the ketoconazole, and you wait 24 hours, just put in fresh media, and you see this nice return to kind of a little bit of a jump, but after 72 hours with no compound, you see a nice return to 100%, or the original starting amount of 3A4, and we did this in 2 donors, 2 human donors. We did the same thing with diltiazem. This is a time-dependent inhibitor, so it takes time to form the inhibition, and you can see that in the data as well. So 0, about 10 minutes in incubation, you see no inhibition at all. After 72 hours of dosing, you see this really nice concentration-dependent curve of inhibition. Now you remove the diltiazem and you allow the cells to recalibrate, and after 24 hours, you're right around here, which is pretty good. At the higher concentrations, it hasn't rebounded as much, but at the lower ones, it does a pretty good job. You're about 80% of what you started with. Then go out for 3 days, 2 days after that, 72 hours of recovery, and you get these huge induction effects, and we thought that was kind of different, not explained, and we haven't seen it in literature. So we tested it in another human, and we got the same results. So it's definitely not a fluke and probably deserves some more attention. And we took a classic inducer, rifampin, PXR activation, and you can see on the graphs here that if you incubate time 0, really no induction. 72 hours after incubation, you're about 2-fold induction. Then you remove the compound, and this is very interesting. What you see is even after 24 hours without the compound, it's still inducing, and you're at about 3-fold induction. Then it's 72 hours, you start to see this time-dependent recovery. So at the higher concentrations, you're still pretty induced, but at the lower ones, you can kind of see it coming back down to normal. And after 120 hours of recovery, you're back at the 100% or normal steady state function before we started the induction. And again, we repeated that in another human. What we've been working on lately in our labs is trying to accurately measure what goes on in the bile, what gets secreted in the bile, because we know that this happens to be an important pathway of elimination. And you have these, 5 major transporters: MRP2, BSEP, PGP, MATE1, and BCRP, and we try to get a method that would really help us predict what's in the bile. So currently, or the way it has been done for many years, is indirect bile measurement. So in one well, you have the bile canaliculus closed, you add your compound, 20 minutes or an hour, whatever incubation time you like. In the other well, you have an open bile canaliculus. You lyse both wells after 20 minutes, and you subtract, and you get what's in the bile. So it's not a direct measurement of what's in the bile. What we did is something a little bit different. We said, all right, we have really good mass spec tools. Why not try to get what's directly in the bile? So we incubate in one well for 20 minutes or an hour. You remove the compound, you open up the canaliculus and get out what's in the bile. Then once you have what's in the bile, you can lyse the cells in the very same well, and you add them up and you get what's in the cells plus the bile. So it's a very direct measurement of what's going on in the bile. Just a couple of slides on the data that we did. So we did torcolate, MRP2, I think, or BSEP, and what you have is you have your cell, your bile, what's in your cell, what's in your bile, and you can get uptake rates, biliary clearance, biliary excretion index, all from these 2 numbers. And then if we compare it to the hepatocytes in double gels and the 2-well method up here, this is a paper by B from Pfizer, they were getting anywhere, the biliary excretion from 41 to 72, depending on the donor, and our donor was about 66 plus or minus. So it's a pretty good correlation of what's been reported in literature. Same thing with estradiol-glucuronide, different transporter, but similar type of data. So you have what's in your cells, what's in your bile. You can add them up, get all these different numbers. And again, in the literature, reported 37 BEI, and we have somewhere from 40 to 43. So again, a pretty good correlation. one of the questions that I asked the guys in the lab is how close is this to in vivo? Because we get that question all the time. They're generating nice in vitro data. How close is it to in vivo? So we found a paper by Lundquist, I think this is AstraZeneca, 2014, where they actually did a rat in vivo study, cannulated the bile duct, and got a rat in vivo clearance, biliary clearance for digoxin and rosuvastatin. So we did it in our labs, used our one well method, got a predicted clearance of 2.5, they got 0.8, so that's a threefold. Rosuvastatin 27, and they got 48, so that's less than twofold, almost one. So again, very good correlation between what's reported in vivo and what we found in vitro. I'm going to skip around now and go a little bit to the tox stuff because we're focused on anything, any place a good hepatocyte can be used, and I think the tox arena is one of these very important areas. We did a study with Sanofi where we measured the toxicity in human on the left, rat and dog, and we did this in a 14-day repeat dosing study. So you change the media every 2 days, add fresh compound, and monitor. This is with ATP. So you can see that after 24 hours, which is this line up here, you see very little toxicity of cyclophosphamide, but go out for 7 or 14 days and you start to see these really nice curves. In the rat, it's even more interesting because you see these left shifts from day 7 to 14, and in the dog you don't. It looks actually pretty similar to human. So very, very good data, interesting data. With Sanofi, I think we ran a total of, 20-something, 29 compounds or something like that. And for it's a busy slide, but what we want to concentrate on here is this top one where these drugs are known to be reactive metabolites or cholestatic mediated responses, and we picked up all of them if you compare 24-hour to 7-day ratios of TC50s or TC50 over Cmax, whatever you really want to look at. And this is something that we think can add to the regular toxicity studies where you have an ATP endpoint and you're looking for something that's under 100 times Cmax or 100 micromolar. This is another tox slide, but an interesting slide because it combines tox with metabolism. So we know that cyclophosphamide is a metabolite-generated toxicity. So we use the ACEA platform on top here. I don't know if you're familiar with the ACEA platform. If you're not, it's a platform that has electrodes on the bottom of a plate, and it measures impedance. So basically cell stickiness and morphology on the plate. And as the cells become more and more toxic or have a toxic response, their morphology changes, they start to come off the plate, round up, and you can really see these differences in impedance. So this is a cell index, just how sticky the cell is, and then this is time, so you kind of know when the toxicity is happening compared to when you put in the drug. So this is dose 1 of the drug on day 0, dose 2 of the drug 2 days later, 3 and 4, and you can see these kind of little spikes where we dose the drug. That's because we take them off the machine, redose in the hood, and then put them back on the machine. And the machine takes readings, I think, every 10 minutes or half an hour or so. And the experiment that we did here was we did cyclophosphamide alone and cyclophosphamide with ABT. So aminobenzotriazole is a cross-CYP inhibitor. It should inhibit most CYPs. And when you add it to the media with the compound, what you should have is CYP inhibition and no toxicity, and that's exactly what we see. So on the bottom here, you have the blue. You can pay attention to the blue and the green. So this is cyclophosphamide with no ABT, and you see these nice curves. The cells are definitely dying. Now you add cyclophosphamide plus the ABT, so that's the light blue and purple, and you see that there's really no cell death at all. Maybe right around day, I guess this is day 6 already, but I think this is just an artifact of the system. And we repeated this with a simple glow assay, so cell titer blue, Promega kit, and again, you see the similar results. Without ABT, you see this toxicity here, so you have maybe 18% of the cells are still alive. Add ABT, and you have almost 100% live cells. So this is a very good proof of concept about how you can really identify metabolic toxicity effects just by using a simple inhibition with ABT. The last couple of slides I'm going to talk about is really what the future looks like for Hurel, and as Les said, what started us off is really a microfluidic device, and we've spent a lot of time engineering a device that's going to be easy to use and very practical and convenient. This is the chip that we currently are testing in our labs now. Each of these has three little devices that you can use, 3 separate experiments with 2 chambers. So it'll be liver plus heart, liver plus kidney, disease model, transfected target. You can really fill it up with whatever you want. You know, the world is our oyster. The chip goes into our device, which you then put in an incubator, and you can run it for anywhere from 3 to 7 days, depending on what you want to do. So a proof of concept here would be if you have liver in one chamber, colon cancer in another chamber. One of the colon cancer drugs that's being used is Tegafur. The prodrug is Tegafur is prodrug, 5-fluorouracil is what really kills the cancers. So what you need is to dose with Tegafur, have a liver or CYP-mediated transformation. You get 5-fluorouracil, and that should kill your cells, your cancer cells. And that's exactly what we found. So if you look, I guess over here, if you have the liver compartment and you add Tegafur, you see this strong toxicity. If you add, here, this is right, you see strong toxicity. If you don't add Tegafur, you don't see, if you don't have the liver compartment, you don't see much at all. Again, proof of principle, and right now we're validating all of this in our labs, and hope to get it to you, the customers, soon. So what are the advantages of using Hurel? We're highly stable, reproducible cellular competency. We have the most cells per well, I think, versus the competition. Long endurance and high function enables many different experiments that we can do, questions we can ask, repeat dosing studies, drug-drug interactions, complete metabolite formation, emphasis on lab practicality and convenience. We airship throughout Western Europe and North America. Plates arrive ready for use. Brief acclimation period, they're good to go. All the standard formats available, and all the media is included. Thank you.